Hi everybody, welcome to the webinar today. Today we're going to be discussing FCS Express 4 Image Cytometry, some special applications, and this is going to be an application example from Er Liu, Ph.D. And Er Liu is from, again, Jolla Bioengineering Institute, and he's going to be discussing the utility of FCS Express software in Fluorescent and Surface enhanced Raman scattering for image cytometry-based data. So I just wanted to start today by telling you a little bit about FCS Express and maybe some of the reasons why Er has actually used the software to great results with his application. FCS Express has been developed as a powerful flow cytometry data analysis and presentation software. And we're proud that we've released our image cytometry product in the last year or two. The main principles of flow cytometry and image cytometry overlap, such as taking multi-channel fluorescence data and plotting that data in scatter plots and histograms to look for statistically significant differences and changes. What we can now do is take your image cytometry data a step further by leveraging the most powerful analytical tools in FCS Express for flow cytometry to analyze your multi-parametric image cytometry results. We can relate these results from the image cytometry experiments from the dot plots and the histograms or any type of plots in FCS Express back to the original image for confirmation of your results, so you can get spatial information, and you can even move from the population-based level from dot plots, scatter plots, down to individual cells in FCS Express. So in the end, FCS Express is going to allow you to spend less time reviewing large, overwhelming data sets and analyzing raw microscopy images by using tools for flow cytometry that have been proven to handle data sets with millions of data points and allowing you access to the raw images when you need. So before we begin, I just wanted to give you a quick overview of the workflow of how data is generated from image cytometry experiments and how that's brought into FCS Express. So first of all, you have an image acquisition step. This is going to be where your images are acquired on a microscope, a confocal. It can even be a digital image, and nowadays you could even take pictures with your cell phone or iPhone, and you can actually get those into FCS Express if you want. The next step is going to be an image segmentation. And image segmentation software packages such as CellProfiler or ImageJ, even packages from molecular devices from their Velos instrument, Chemometic, Nexlam, these are a few packages that we are currently compatible with. And the image segmentation is going to allow you to define cell boundaries, define where an object is, define where something is in the picture. And what we can do, or what CellProfiler or ImageJ or any of these packages can do, is then extract all of the features from those individual objects or cells. Features such as intensities and areas are extracted from the image, and FCS Express will be able to handle as many features or parameters as this third-party segmentation software can produce. Once you have all the images segmented, you have the features extracted, you can then export that data directly to FCS Express for data analysis. We can generate very nice reports from that data, and we communicate and publish those results as well. And what Er is going to show you today is some data that he's been acquiring. I believe he's doing image segmentation with CellProfiler, and he's taking the CellProfiler data and bringing that directly into FCS Express, where he's been generating some very nice reports, some interesting data sets, and it looks like right now he's ready to communicate those results to the rest of us here on the webinar. So with no further ado, I'd like to introduce Er Liu from Jolla Bioengineering Institute. Er, I'd like to turn the presentation over to you, and you can discuss what's going on with your data analysis.
All right. Thank you for your introduction. So it's a great pleasure to be here to share with you my research on the surface-enhanced Raman scattering-based spectral imaging and cytometry using the FCS Express software tools. So first of all, I'll give a brief introduction of what I do in the lab, and later I'll talk about the case studies using the software tools from the Novo software. And finally, I'll give a summary and acknowledgements. So to begin with, cancer. Cancer is the leading cause of death nowadays, and especially breast cancer is the top incidence in the world. And unfortunately, it's still a big question for scientists and medical experts to solve. It's not one single disease. It's very heterogeneous. So people usually use breast cancer cell lines derived from the breast cancer patient as a model system to study the biology of the cancer itself. So in this case, for breast cancer, there are more than 50 breast cancer cell lines derived from the patients. And traditional gold standard to identify those different subtypes are basically based on the estrogen receptor, progesterone receptor, and HER2 expression. So this is a pretty rough classification. And in order to further identify the subtypes, usually we use a panel of protein markers, as you can see from this table. So I will give you an example of a couple of subtypes of breast cancer cell lines. In this case, there are 4 different breast cancer cell lines representing different subtypes like basal, luminal, and interestingly, for traditional gold standard markers, there's very limited information out there. So we have to look for more. In this case, we're looking for keratin, EGFR, CD44, CD24, CD61, CD29. So we need a panel of markers. So naturally, one of the questions that come to mind is how can we stain the cells or look at the different cell lines that are stained with 12 markers? Is it possible? And naturally, we are looking at fluorescence staining. In image cytometry, we know that the hardware is one of the limiting factors to look at 12, for example, 12 channels at one time. Usually a well-designed microscopy system, you can have 4 channels listed here, as you can see from the DAPI, GFP, Sci3, and Sci5. So this is largely because of the fluorescence itself. It's a broad bandwidth. So you can't afford more than 4, I would say. If you carefully design it, maybe we can have something like 6 or 7 channels at a time, but it can't go more than that. And on the other hand, we can think about Raman tags as a replacement for the fluorescence. So basically, the Raman signal is based from the scattering properties, and it has very narrow bandwidth. And the scattering intensity changes as the laser excitation changes. And one of the questions for the Raman scattering is the signals is very weak. And unfortunately, our group has been working with surface-enhanced Raman scattering, where we got very good signals that are comparable to the fluorescence. So the way fluorescence imaging and the Raman imaging are implemented are relatively different. So for Raman, we only need very simple instruments. On the other hand, for fluorescence imaging, we need a different filter cubes to select, and we need a bunch of cameras to capture. So Raman definitely have some advantages over fluorescence. And if you want to see more about the Raman tags that we developed in the lab, there are a bunch of references listed here. So our objective is to design a SERS, bright field, and fluorescent multimodal imaging system for cytometry. And we want to look at multiplex single-cell analysis in cancer, stem cells, and drug screening. So the multimodal imaging instrumentation was implemented, and if you can see here, so first we put down the cells onto a regular cover slip or multi-well plate, and we fix and stain with either fluorescent tags or SERS tags. So after that, we put them into a microscope with a motorized stage. And we use one CCD camera in combination with filter sets to get fluorescent images. Usually we stain the cells with DAPI for nuclei identification and actin for whole cell identification, as well as another protein marker. On the other hand, for SERS imaging, for the Raman signal collection, we're using a 633 nanometer laser and using point scanning with the help of our motorized stage, and the spectral data were collected and filtered through our spectrograph and a notch filter. And using the CCD camera, we can get a series of spectral data for each point that we scanned. So all of this are implemented in Micromanager, and here is one example of the picture of our actual instrumentation. So after we collected all the spectral data, the next step is to reconstruct the spectral into image. So essentially, when we perform the scanning, we actually acquire a 3D image. It's X, Y, and lambda, which is the Raman shift. So for each pixel, each voxel, you will see it's a spectral that looks like this. So the very first step is to create an interpolation of all the spectral during the Raman channel. So we call it interpolation. But similar to the 3D imaging X1Z, you got a projection image. So this will tell you how much signal you have, and hopefully you will see the whole cell, but you don't know which marker you have. Therefore, using a spectral unmixing technique, which is a procedure that measures the spectral of mixed pixel and decompose it into different components. We also call them end members, and reconstruct them into images of each SERS channel, as you can see here. I color-coded at different colors just for identifications. So after we acquire the image reconstruction, we have fluorescent channels, images from fluorescent channels, images from SERS interpolation. We also have the unmixed images of each individual SERS channel. Then we'll go through first the registration process, because all the images are acquired from different imaging modalities. So we have to do registration first. So all of these steps are implemented in ImageJ or Fiji. So after that, we have fluorescent channels, we have SERS channels. For the fluorescent channels, we use it for mask creation. So first we go through a bunch of filtering process, background subtraction, to segment out the cell nuclei, the whole cell, and the subcellular compartment. And after that, we basically superimpose our nuclei, whole cell, and subcellular compartment mask to the SERS channels and make the morphology and intensity measurement to create metrics from each individual cell. So these are implemented in CellProfiler, and we'll export it to FCS Express Image. So combining our raw images together with the numerical data generated from CellProfiler, we can generate reports and perform exploratory data analysis in FCS Express. So next, I'll give you some information about how we did it in our actual cell studies. So here is how we set up the experiment. So first, we want to do the analysis in a high throughput and high content manner. So we're having 4 cell lines from different subtypes. See the cells to a 96 well plate. And you will see for each row we present the same cell line from A to B, and from C to D, that's another cell line. And for each column, we plan to stain each column with a different either a surface marker or protein marker of interest. So theoretically, we can stain the whole 96 well plate with 12 different markers and 4 different cell lines with a duplicate. So first, we fix and stain with surface markers, 6 surface markers, but in this case we're looking at the fluorescent staining. And using FCS Express and CellProfiler, we perform single-cell analysis to get the cytometry data. And in parallel, we also perform the surface marker staining with our SERS tags for the other half and perform similar analysis. And you want to see if we get similar results from our SERS staining over our fluorescent staining. So that's the very first question we want to ask ourselves. So this is one example of images for one single marker staining, the CD44 staining, with fluorescent tags. So essentially, for each column, you'll see there are different cell lines: MB231, MCF7, BT474, and MB435. And we are staining with nuclei, actin, and the CD44. So here, for CD44, we're using a Dye-Lite 649 gold anti-mouse IgG antibody. And supposedly, CD44 is positive for basal cell subtype, in this case MB231 and MB435. And you can see qualitatively they are expressing. And for the other two, they are luminal subtype, and you won't see much of signal. And similarly, if you look at the SERS staining, again, we stain the cells with nuclei, actin, as well as CD44 with a SERS gold anti-mouse IgG antibody. And qualitatively, you will see that the MB231 and MB435 cells are positive, and for the other two cell types, they are not. So having acquired those images, what's next? We're using our image analysis pipelines. So first, we have to read these images into CellProfiler. And to note, we're expecting to do high content and high throughput. Therefore, there are special requirements for image file names. So we have to design or rename our image files to be compatible for the multi-well plate format, as you can see here. So after we read an image, we have to perform some kind of background subtraction. And here we are using correct illumination modules from CellProfiler to correct the illumination for 3 channels, for the dapiactin as well as our 649, the far right channel. And after that, we have to save the images after correction, and later we'll perform segmentation and mask creation. So after that, we'll perform the single cell feature extraction on the morphology as well as intensity. And finally, we'll export our results to FCS Express compatible format. So after we're reading the image, this one example, we perform background subtraction. As you can see, the blue here, this is the blue channel after subtraction and before subtraction, and the green channel as well as the red channels. And the nuclei segmentation always comes first. So there are a bunch of thresholding methodology available out there in CellProfiler, and it's here I'm using O2 adaptive, and you can choose other methods depending on your applications. And I have to fine-tune a little bit about those parameters that I'm using. And here you will see a panel of original image, the identified nuclei, and the outlines. And there are a few statistics over here that you can visualize during this pipeline cycle. And later on, using the nuclei as a seed, we will get the whole cell segmentation. And again, depending on your applications, you can pick different methods for the whole cell segmentation process. And after we have the whole cell segmented out, we can perform a mask subtraction process to create the intracellular or the subcellular compartment as another mask, as you can see here. So the nuclei, whole cell, and the cytoplasm or subcellular compartments in this case. So after that, we'll measure the single cell features and save the images and the data to FCS Express compatible format, where you have a MATLAB output, you have a CellProfiler output, and you have the mask or the labels from nuclei and whole cell, as well as the cytoplasm. And in particular, I'd like to point out that because I'm trying to do a high content and high throughput analysis, we'll have to throw in an experiment.txt file specifying which well plate that I use, which is critical for us or for the CellProfiler to communicate with FCS Express. So after that, we'll read it in FCS Express. So this is the one of the layouts that I created for our experimental results for indirect SERS staining. So this is the first page of the layout. And naturally, when we perform high throughput screening or high count screening, we want to see the overview of our well data first. So this is one example, and we're visualizing the mean pixel intensity of each well, the expression levels of each protein markers on different cell lines. And if we want to do further analysis, we'll have to generate gates, obviously, like what we do in flow. So here is the example of a well gate setup. So basically, we're setting up each column to be one first level gate, the primary gate, as you can see here. And after that, we'll set up each row as cell gates, as you can see here. And when we are combining the protein marker with the cells, we'll create the secondary gates, as you can see here. So for each well, you'll see each individual secondary gates. And like what we do in flow, we can look at the cell size if we plot the cell area versus perimeter. And similarly, we can also visualize the cells versus the marking intensity or the expression with two different parameters, integral emission or the mean pixel intensity. And for the first experiment, we also want to see if there are any differences in terms of our isotype controls. So we're also here to visualize the isotype controls from different cell lines. So after that, we'll go to each individual biomarker gates that we created. So this is the active gate. It's a CD44 gate. And in this region is the tile scan image collage of each well gates from different cell type, from BT474, MB231, MCF7, and MB435. And similarly, the cell size and expression versus cell size plots are included in this page. And the most important is the third page, where we'll see a histogram of all the conditions at a given biomarker gate, in this case CD44. We plot them all together in one gate and in one histogram, and you will see the statistics over there. And you will see that the median of MB231 and MB435 are a lot higher than the others, indicating the positive staining. And if we look at further histograms for individual cell type. And we can also see that the individual cell staining of each cell type versus the isotype, respective isotype controls over here. You'll see clearly that the two cell types that are supposed to be positive, they are positive, and there are two negative cell lines as well. Similarly, we can create another layout for the other protein markers, in this case HER2. The active gates are represented here, and the reports are generated here. So, similar single-cell cytometry can also be performed on the fluorescent staining channel, where we'll get similar histogram here. And combining our SERS staining results here with our fluorescent staining results, we come to a plot here. If we plot the mean pixel intensity from the SERS staining versus fluorescent staining, you'll see that they get pretty good correlations, which validate that the SERS staining can work as an alternative to fluorescent staining. So after the validation of our SERS staining, what we are interested in is the multiplexing. So this is a traditional paradigm for surface marker staining. We have to stain the cells individually with 1 or 2 channels. So we can stain 12 channels in 96 well plate for fluorescent staining. And for SERS staining, we want to do 12 surface marker staining in one cell type, instead of staining each individual cells one at a time. We want to stain them all together and see if we can distinguish the 12 different SERS tags. So in order to implement that, we first try our capture bead experiment before going to the cell experiment. So here is the results. So each column, you'll see that's a bright field image of image tile scan for each single stained SERS tags on the capture bead. And well, on the second column is the SERS integrated emission image of single stained bead. And the third column to the very last column, you'll see that's the unmixed reconstructed image of each single SERS channel. And you'll see like each row represents the single stained SERS beads. So very interestingly, we can see that images definitely show that they can be well separated. And if we look at cytometry type data, the histogram of those SERS tags staining, and they behave pretty well there, except a couple of exceptions. They can be baseline resolved from all the other channels, as you can see one here. So in summary, here we have demonstrated a multimodal imaging system to perform Raman, bright field, and fluorescent imaging. And we have demonstrated our SERS and fluorescent staining validation results, as well as our preliminary multiplexing data using the software tools from CellProfiler and FCS Express. And finally, I'd like to acknowledge the whole Nolan group at La Jolla Bioengineering Institute, and I'd like to acknowledge our collaborators from Scripps Institute and the UCSD, as well as the micromanager that support our data acquisition, as well as CellProfiler, FCS Express from the Novo software, and Imagine Fiji. And finally, our funding source, the Bioengineering Research Partnership. And finally, I'd like to take any questions. Thank you.
Thank you very much, Er. I'd like to open up the floor for any questions. If there's any questions anybody has, please find the questions and answers box in the GoToMeeting panel, and you can go ahead and type your questions there. So while questions are coming in, I'd like to summarize a little bit about FCS Express. We saw a lot of the work that Er is doing in FCS Express and how exciting that is. But just so you know a little bit more, FCS Express is going to leverage all the most powerful and proven tools of flow cytometry analysis with a lot of new features to improve your image cytometry analysis. You saw how Er was using histograms and markers and gates to actually look at positive populations in an objective manner. This is something we can easily do with FCS Express. One thing that you didn't see is that we can move from the population-based level down to a single cell as well. At any time through a click of just a few buttons. And you also saw that with our new image handling features, you can get full interactivity between all data plots and the images themselves. And when you're ready to go down to the single cell level, we can use our cell galleries. So all of this data analysis, plotting, visualization, the advanced report generation tools, these are all available within FCS Express. We also have many collaboration features and tools within the software, including what we call a free reader, which you can learn more about at the Novo software website. And an intuitive Microsoft PowerPoint-like feel, you can see that we're actually within FCS Express right now, and it looks like PowerPoint. This is going to make the software very easy to use and understand for any level of user. You'll also notice on our website and within FCS Express that we have many resources for our customers. There's tutorials, sample data, and an online forum. The tutorials and sample data will cover working with CellProfiler, working with picture plots, working with high content analysis. All of the basic things that you saw Er working on today can be done through these tutorials, and then you can take them to a more advanced level like Er has done on his own. If you'd like to try FCS Express, you can certainly get a free trial from our website at www.denovosoftware.com. We also have a great support system. If you have any questions regarding FCS Express, regarding today's webinar, any questions either for Novo software, or if you'd like to relay our questions to Er Liu, you can reach us at support@denovosoftware.com. And I also wanted to end by just bringing up the Novo software website. If at any time you'd like to get a download, you can come here and get our free demo. You'll also notice on our main page that we have some nice videos highlighting everything available within FCS Express, as well as a webinar section listing all upcoming webinars, and a recorded webinar section where you can come back to and review any of the webinars that we have available, including the webinar from today that's going to be posted on the recorded webinars page. So Er, thanks again for your presentation today. It was great. If anybody has any questions again for Er, please feel free to contact us at support@denovosoftware.com, and we'd be happy to send those questions over to him. Er, do you have any more comments today before we end the webinar?
One of the comments that I have is the FCS Express images are really interesting and useful tool for me, because I'm the application scientist who is trying to put all the tools together to do cell imaging, analysis, and data mining together. So this is very useful for me, and I will be really happy to see the improvement of the image cytometry in addition to the flow cytometry.